Good evening. I'm John Sprumberg, the president of the CUNY Graduate Center, and it's my pleasure to welcome you to tonight's conversation with Nobel laureate Dr. Katalin Karikó and New York Times writer Carl Zimmer. We are thrilled to hold this event, part of our City of Science series, once again in partnership with Cold Spring Harbor Laboratory and its BGI Nobel Laureate Program. Tonight we'll hear from a brilliant, tenacious woman scientist about her search for a solution to a vexing problem and her against-the-odds success. Her inspiring story, like many Nobel Prize-winning scientists, exemplifies the values we at the Graduate Center hold dear. These include the pursuit of excellence, the importance of scientific discovery, and the notion that research can advance the public good. For more than 60 years, the Graduate Center has been the home of most of the doctoral programs at CUNY, the City University of New York. We currently offer 49 doctoral and master's programs spanning the academic alphabet from astrophysics to urban education. We need to get to zoology one day. Our scholars include winners of the Nobel Prize, the Nobel Prize in Mathematics, and the Pulitzer Prize. In classrooms and labs in this building and uptown at our Advanced Science Research Center, our students and faculty engage in the rigorous research and collaborative interdisciplinary learning that sparks innovation and leads to a more humane and just society. We are proud of our history of offering free public programs like this evening's event that brings excitement from the classroom to this stage. I thank all of you for being here tonight and our organizers for making this event possible. I want to note you should have got a card, and if you didn't get one, our ushers will be around later with a card. And this being a school, I have to tell you, penmanship counts. If you want your question to be picked or potentially picked, it has to be able to be read. So please write in block letters and make it big. It is now my pleasure to welcome Lamila Pollock, the Executive Director of the Library and Archives at Cold Spring Harbor Laboratory, who will introduce our guest. Mila?
Thank you, Josh, and thank you for coming to this seems wonderful event, and I'm sure that you would agree with me after the event will be over. I'm so pleased to co-host this annual lecture with the CUNY Graduate Center. This is the third event that we are doing together, Cold Spring Harbor Laboratory and CUNY. It is such an honor to bring remarkable researchers, including like this evening, Nobel Prize laureate to New York, and to provide an opportunity for them to tell us about their work to make our society better, and to give you an opportunity to interact with them. A few words about our Cold Spring Harbor Laboratory. We're a private, not-for-profit laboratory. Bruce Tillman is our CEO and president, and Marilyn Simon is chair of our board of trustees, who is with us today. Thank you, Marilyn, to join us. The lab has been home to 8 Nobel Prize winners. We have research programs in cancer, neuroscience, plant biology, quantitative biology with over 600 scientists. We have many educational programs for students, including high school students, and including the DNA Learning Center at CUNY City Tech in Brooklyn, another collaboration with CUNY. Our over 12,000 scientists coming to our international meetings and courses yearly here, and also at our conference facility in China. Our Center for the Humanity and History of Human Biology provides research facilities and support for visiting scientists. They come to use our world-renowned archives, which contains the collections of many eminent scientists. We also hold events such as exhibitions, lectures. We hold the events in our place on Long Island, and many of them we have on Zoom or after that on YouTube. So you are welcome. If you miss them, you're welcome to come and to watch and to listen to their fantastic events. I want to thank BGI Genomics for their incredible support for our Nobel Prize laureate archives program. A representative of BGI, Charles Bao from Complete Genomics, is here today. And Charles, thank you very much for your wonderful, incredible support of this program. It is now my pleasure to introduce Katalin Karikó. Kati is the winner of the 2023 Nobel Prize in Physiology and Medicine and the 2022 Breakthrough Prize in Life Sciences. Her work on RNA was the foundation of the rapid development of effective COVID-19 vaccines, for which she won the Nobel Prize. Dr. Karikó's research has a broader impact for the development of other RNA-based gene therapy. Dr. Karikó is a professor at the University of Szeged, Hungary, and adjunct professor at the University of Pennsylvania. Our another guest is Carl Zimmer. I don't think that he needs the introduction, but I have to tell you. Carl is a writer, and he writes the original column for the New York Times, a regular contributor to NPR Radio Lab, and an adjunct professor in the Department of Molecular Biophysics and Biochemistry at Yale University. Carl Zimmer has written many books and has received a number of awards, including the Stephen Jay Gould Prize and the National Academies of Science Communication Award. His forthcoming book is Air-Borne, The Hidden History of the Life We Breathe. We can read in this year, correct? It will be published in February. Welcome, and the floor is yours.
Okay, well, thank you so much for coming out. Thanks to everyone online. I'm very much looking forward to having a conversation with you about your life and work. But you know, I should preface this by saying I had a kind of a cheat sheet. Katy actually wrote a whole memoir. And it's really quite remarkable. It's called Breaking Through. And so if you haven't read it, I highly recommend that you get it, because it's got science, it's got Cold War history. It's a very deeply personal story about Katy and her husband and daughter making a new life for themselves in the United States. This spoiler alert, this came out before she got the Nobel Prize. So if you're just waiting for that big climax at the end, it's, at least in this edition, it's not there. So I have a sort of a writer to writer. I have a question. What sticks out for you from going to Stockholm to get the Nobel Prize? What surprising thing would you put like in an epilogue to your book?
Okay, so I mean, it is very special to go to Stockholm. I have never been in Stockholm before, so that was my first trip.
Well, we could all go to Stockholm. but you went to Stockholm.
Yes. Yeah, and meeting the king and sitting next to the prime minister. I mean, this is unreal for a person like I am. So I was not in the spotlight in my life, not like that. So it was, you know, I like quiet place in the laboratory and not in the, you know, asking all of these reports and going to the, private audience. I thought that, just the king and me, and then 150 other guests were there. So it was like, so it was very, very special. Just, I always was thinking about other people who were there and who get this award. And I never thought myself that I always mentioned that, in the name of many other scientists, many other honest working scientists who is not recognized in their name, I accept.
Mm-hmm. So in your memoir, you start with a really searing image, a memory from your childhood. You are staring at a dead pig. So tell us about that moment and why you chose that as where you wanted to start telling your life.
I was like 3-4 years old. I did not remember. My parents said that I was, my father was butcher, and then every family had one pig, and then when he opened it, I want to see, I was standing there, what is inside, which made this animal running, and I had an older sister, 3 years older. Later, she became an economist and got a PhD in economics. She was far away inside the house. She never wanted to see what is inside. So I was the one, and they told me that I was so curious, want to see everything what's happening. And that's...
So do you think that curiosity that would make you stare inside of a dead pig would lead you to into science? Do you see a connection there?
Yeah, and other things. So, that we didn't have cows, but neighbors had. And I have seen when it gave birth, how they pulled out the animal. And I like to climb up the trees. It's a small town, and you do these things. And I was just like to see the birds, the nest, and now the little birds are there. So I was fascinated with everything which was around us, so that, we also had a little garden with my sister. That's the worst part of the garden. My parents said, okay, it can be yours. And we put the seeds, and then we were waiting the plants to come out, and all of these things. So it was, part of our life was, watching all of these things in nature around us.
So, as a girl growing up in Hungary at that time, you're, difficult circumstances. Your father lost his job as a butcher because he had organized a local watch when the Soviets arrived. So in all of that, do you feel like you got encouragement to become a scientist, or was that just something that you just pushed through on your own?
The teachers were great, and then we went, like biology class, elementary school. We just went to see the trees, and the teacher just picked up the leaves. Oh, it is yellow. Was it the green became yellow? Or, that he asked questions, and we didn't get the answer. And, there were no Googles in the 60s. So that, you just wonder that, the color was there, and the green covered it, or the green, all of these things. So the education was such. And, yes, indeed, my father did odd jobs, but, he was always happy and, whistling, singing, always. And so I did not notice these things. And so many things we didn't have. This was an adobe house. We lived 4 of us in one room. We had no refrigerator, no television, but nobody had. I didn't know that I don't have these things. And we had a dirt road, and, the cows went to the field every morning very early, and then in the evening you could see smart cows coming. Each knew that which house had to stop by, and you just watch these things around. And so it's a very simple life, but what was there is love, and, that's what most important. I didn't see politics, you just live in a family.
Mm-hmm. You do mention memories about polio, and that polio was a real threat when you were young and you would be vaccinated. Was that make it stick with you? I mean, you obviously
Yes.
you didn't decide you wanted to make vaccines then and there, but like it seems like it was a pretty strong memory.
Even later, I never wanted to make vaccine. I want to make mRNA coding for a therapeutic protein, and I imagine that people will have in their freezer some kind of mRNA, and then they are cooking and burning hands, and they said, oh, where is the collagen 7 to put mRNA there and put it there, and okay, now it was healing or something like that. So that's what I imagine. But, I remember also then we had the chickens, and, we vaccinated that. And so we never questioned. It was, we were happy that, the animals will be healthy.
Mm-hmm.
And I remember that who had cows, this is a tuberculosis-free cows, was on the building, so we could go and purchase the milk.
So you went to University of Szeged. Did I get that, pronounce that right?
Yes.
Okay. Hungarian is not my strong suit. But in any case, you're going to University of Szeged. And so there, we can sort of see the first steps you're taking towards the kind of research that you were going to pursue single-mindedly here in the United States. I mean, look, maybe we should just in a way, like it's a sort of foreshadowing of the mRNA vaccines, because mRNA vaccines, they have a lipid coating, a shell, and inside there's a messenger RNA. And you started to work with lipids and with RNA when you went to the university. Tell us about sort of how you started to get to work with those things.
So, when I went to the university, the first day we had to make a statement what we like. I said, I like plants. And everybody thought that, oh, Kati, it is so boring. Because everybody was for genetics. And so I went there because University of Szeged is in Szeged, and the same year when I started in 1973, they opened the Biological Research Center, and that was my dream, that I will have one day a job there, and I will work every day. I discover some new things. I have no idea how scientists, what they are doing, but I thought some vitamin I might discover or something. And then the only opening was in a lipid team, kind of boring, yeah? That's really boring. But I thought that, okay, I will go there in the summer and as a student. And the head of the labs told me that, oh, Kati, you will have the rest of your life to work at the bench. Go to this fishery institute and for
fisheries.
Fisheries, yeah.
So
and then
fish oil and
fish, and then take samples from the fish because it's farm fish and some water fleas collect. And you dream about something, and finally you are in the pod there and collecting little insects, and so you just go with the flow, and I went there, and at that point, actually, when he told me, I didn't know, I am 58 years old, I still do all of the experiment with my own hands, I pour the gel, culture, everything. I didn't know, when he said that rest of your life, that, all of my life, I will be at the bench. And so it was, so I went there in the summer, and then, when I came back, and later I did several years I working there. One day, the guy who had in the RNA unit, he just walked in and he said, oh, I have an opening. Do you want to work with RNA? I said, oh, okay. I am not a visionary who would see, okay, now it's lipid, we have to wrap up the RNA. And I didn't know any of these. I just went with the flow, and that the RNA was a short RNA, and it had an antiviral effect that was supposed to that.
So this was a
little piece of RNA. I had to synthesize, and I had to set up a screening lab where, with viruses, I will test out whether it can inhibit the virus. This little piece of RNA.
little piece of RNA that could inhibit viruses by triggering cells to produce an immune effect. Is that the idea?
It will activate some enzyme and end of it, cut up the viral RNA. That was the idea. And so that little molecule was, little RNA was discovered just 1977, and I started my PhD in 78, and we need an antiviral compound. That was, my supervisor said, Jenny Thomas, and then I said, okay, antiviral compound, even today we need one, a good one, but, that's what in day one, when I work even in basic science, it was there that it has to be useful for something.
So you're working at Szeged trying to make this virus-fighting RNA treatment, and you actually, this is the time when you meet your husband and you start your family, and then suddenly it's over. I mean, it sounds like it was really abrupt that whole project just ended.
Yeah. So the company sponsored this program, and then on my 30th birthday, I learned that I have to find another job. That's how it happened. I have to say that I was straight A. I was valedictorian in high school also, and in elementary school I already was third best in the country in biology competition. So I was kind of good, and all of a sudden, okay, I'm not that good.
Oh man. There were not a lot of jobs for people who work with lipids and RNA in Hungary. I mean, you needed to be looking elsewhere.
Yeah, I tried to apply, not even today, they never responded. And I applied in Europe, who did a similar small molecule with this antiviral effect. Everybody responded, and they said, just bring the money with your salary. But, we are behind the Iron Curtain, so we were not eligible to apply for fund. For to go to London, Montpellier, or Madrid. That was the 3 jobs. And finally, I realized that, oh my God, I have to go to America. I never wanted to come here. My colleagues, were telling me that, Caroline, that, and I knew that New York, I knew that where it is, and California and something, but what is in between, I never cared. Anyway, so.
And I mean, did you know much English?
English was.
Did you know much English when
no,
you moved? So you had to do that too.
Catch up always, so that a very small town. So I went to the university already. I catching up because others already could speak English. Knew that how to work pipette or all of this, chemistry lab. I have never been in chemistry lab. So then again, I started to learn English when I was 18. So this is the ceiling and that's the wall. That was the 2 things I learned the first day. Yeah, so but by the time I got here, I could speak.
Okay. You might be a little tired of this story, but I hope you'll indulge us. It's about the teddy bear. So you're coming to America, and it's kind of difficult to get here from behind the Iron Curtain.
So 1985, is in Hungarian currency was not transferable, and in Hungary you are not allowed to own dollars or any kind of Deutschmark or something. You are not allowed. If somebody gave you, you have to go to the bank and they will give you the corresponding forint. So they said that, my husband can have $50. My daughter was 2.5 years old. She can get $50 to come in America, but I'm not eligible. They said that because I have a job offer at Temple University, so ask them to send me money. I mean, they wouldn't send me. So anyway, so $100 coming with a child, what I will do for 1 month before I getting my first little paycheck. So that we had a car and we sold, and then we had Arab students studying at the medical school, and they had currency, and in black market, I exchanged that, and it was about $1,000 equivalent. It was English pounds, so I had to take a day off to go to inner city to even exchange back to dollar. I didn't know that in America, in the corner bank is not carrying all of these currencies, not like in Europe anyway. So what, it was illegal to own money, such. And so I had to open my daughter's teddy bear my mom gave her, and then I opened the back, and then I put the money in, wrapped it up, and then sew it back, and we gave my daughter the bear. So she smuggled, not us. We kept our eye on, that she won't leave the bear in the airport.
It wasn't me. She's the one with the teddy bear.
Oh.
So she just sort of like carried the teddy bear through the airport.
She carried an airport.
Hiding in plain sight.
Yes. We just could open up, the bear again at the aeroplane.
So you get to America with the teddy bear full of cash, and you start a life here. You start at Temple University, and eventually, the way you describe it is that you got this. In the years that followed in Philadelphia, you got the reputation as the crazy mRNA lady. And tell us how you became known as how did you achieve the title of crazy mRNA lady?
So I worked in RNA, synthesized chemically, enzymatically in Hungary, also here at Temple. At Temple University, we needed an antiviral compound, and we couldn't use this small molecule, but we did a long double-stranded RNA, which was inducing some antiviral activity, interferon, the name is that. So we did run clinical trial, actually, at Temple University, and we treated HIV-infected patients with this RNA, IV injected. And so it was always RNA what I was doing, but at actually 89, when I worked at University of Pennsylvania, that was really messenger RNA I was, doing, and convinced my colleague who hired me for a job that was called research assistant professor. Those only, alien is taking because, it's not tenured position, but you can get a green card, and I needed one. And so I started to work with RNA,
and we should just clarify, so there are lots of different kinds of RNA, and so messenger RNA is what?
Messenger RNA is a type of RNA which codes for a protein. And, we are made of protein, or the protein is an enzyme and makes lipids or something, and that's what we are made of. So, but originally we need, a protein. And yes, and I was reading about all of these antisense stories, and I thought, it would be simpler to just make the mRNA and, would produce a protein which is beneficial, and that would be so great. And already at that point, it was like 1989, 88, already solved the problem that you can make in a tube. You can make an RNA what you like to do. And that's actually how not in that one, but they are doing today also for many other applications. So that's it.
So the genes are in the DNA. Our cells are making messenger RNA from those genes all the time, and then those messenger RNA molecules are where our cells are using them to make proteins. So your idea is, let's put some mRNA, put it into the cell, like in a lipid shell, and then the cell will just make that protein.
Yeah, so the patient could make their own medicine.
Making our own medicine in our cells.
That's what the idea is. But people felt sorry when I mentioned them. Oh, I work with RNA. Oh, poor Kati, this mRNA. Oh, I hate it. Because it always degrades. I hate,
it degrades.
Yeah, because the basic workhorse of molecular biology is actually working with plasmid. And plasmid is, we are making the mRNA from. And then
so a plasmid is a ring of DNA. You're over here working on the mRNA.
And you isolate the plasmid from bacteria, and to remove the bacterial RNA, you add RNase, an enzyme which will cut up the RNA. And the students in the lab, they contaminate everything. the refrigerator door, the jail apparatus, and the people try to analyze RNA and always degrade. And because if you have an RNA and you cook it and boil it, whatever, and then you let it cool this liquid, it comes back. It is all active again, so it is impossible. You have to demolish the building or something to get rid of. But that's what the reason why people couldn't work with RNA. That's what I believe.
So you insisting on working on RNA when everyone's feeling sorry for you, maybe that was why they started calling you the crazy mRNA lady? I mean, because you were just so determined?
Yes, and then because I never get a R01 grant, and they thought that it's a crazy idea, and sometimes they talk behind me. Sometimes I heard, but I didn't care. I found that I can work with it, and it was at the beginning small amount of protein was made from this RNA, short period of time, like kind of useless. But as time goes by, improvements I made, and I was. People consider me very unsuccessful, but I felt that, I solved today that problem, and then I felt very successful. So
you were you were actually able to you put mRNA into cells, and you were getting them to make proteins.
Yes.
And
I thought that this would be useful for something. I just had to figure out what kind of disease you need. At that point, maybe a small amount of protein would make a big difference.
Right, and clearly there were other people who were not appreciating that. Because like at the University of Pennsylvania, you were demoted. I mean, like it was just, you were, it was difficult.
Yes, you can say that. It was.
I'll say that.
So we didn't even say that from Temple University, I was subject for deportation because my supervisor wanted to keep me there, and then also from when I was 40 years old, I was demoted from this faculty position. It was research assistant professor, which I meant just
Right.
not
So
really.
just to recap, left Hungary smuggling $1,000, almost deported, working at Temple, working at Penn and being called the crazy mRNA lady, then demoted. That's a lot.
Yeah. Now I'm 40 years old. And
then you're
went
40,
through.
right. So then, in 1997, you meet the scientist who will be your co-winner of the Nobel Prize, Drew Weissman. How did you two meet?
Yeah, so there was an equipment called Xerox machine or copy machine, I'm just saying for the young one here, that
Once upon a time, we would put paper on glass, and the bright light would flash.
Copy all
the
of
kids,
this.
they don't know about Fox.
Yeah. And so because, and then you put in a, cabinet, and you took out, and you were reading. So that's, and then Drew Weissman came. I already was 10 years working with mRNA, and then this guy was using the machine. Actually, I work already in neurosurgery, but I knew the code, to copy at the Department of Medicine, because where I used to work in cardiology, so I sneaked back, and this guy was there, and he was using, and I was talkative, bragging. I said, I work with RNA, while he was copying there, and then he said that, I came from Fauci's lab. I didn't know who was Fauci. I was not in that field, and there was, not the television at that time. I would know that who is. And he said, I want to develop vaccine prophylactic or therapeutic for HIV treatment or prevention. And I said, I make RNA, and I can make RNA for you. And so that's how we started.
So this was this the first time that you thought about the possibility of, I should back up and say you had been thinking about using mRNA as therapy, as treatment for strokes or things like that. Now, Drew Weissman is talking about vaccines. Had you thought about vaccines before?
No.
So
I was just therapeutic, and that's why I never worried about the RNA or the protein is some kind of foreign, because I just wanted to have a protein which is not the right place, not enough, and I just want the natural human protein to be there, heal the wound or something, and was not thinking about, and I have to say, the immunology, I learned in school, but not because in Hungary was behind, I learned the latest, but by the time I met Drew, like 10 years after I finished schooling, all of this immunology was changed. I learned from Drew that how immunology works. So we educated each other. I taught him about RNA. That's my major topic. But I learned all of this immunology. And then, I understood that, vaccine and I try to have, but I am not a vaccine expert, no. I know one or two things about the mRNA.
Okay, one or two. Sure. But you did discover, working with Dr. Weissman, that there was a problem with this idea of using mRNA as a vaccine, and that there would be this, you were finding there was an immune response that was interfering with it working in mice.
Actually, human cells, what we found first, that Drew made this very special, they called dendritic cells. And these are the professional antigen presenters. These are the most smartest immune cells. And he could make it. I didn't even know what dendritic cells was, I have to say. So, you are not behind. But at that point, I didn't know. Now I know. But that's what, So we found that inflammatory molecules were produced, and Drew was not very much worried about. He thought maybe it's still okay for vaccine or something, but I want to make the mRNA for treating a stroke patient. That's what I try to develop, and I said, it cannot be inflammatory, and so we didn't set out that we will make non-inflammatory RNA. We just wonder why, and then we did a series of experiments. Is all of the RNA is such? And then we just discovered that some RNA was not inflammatory. Those type of RNA called transfer RNA, they had all of this modification in it. And so we thought, maybe we have to introduce these modification to the RNA. But how to do it? Because we scientists always do something nobody did. We don't even know it is possible. And then we just keep trying and believing and trying again. And so that's how it happened.
So you figure out how to get around this immune problem, and then you start to do you tinker more with it, and you're able to get cells to produce more of the protein you want, longer periods of time, and
without inflammation.
Without inflammation, you send a paper to Nature, they reject it, and then in 2013, someone tells you you're not. What is the phrase? Not enough money per square foot, or something like that.
So that
you weren't getting enough grants.
I didn't bring in the money. That was 2013.
Yeah.
So that I couldn't get an R01 grant if somebody's scientist here. This means that you are a scientist in America. An independent scientist. I never get one. So there is hope if you never get one. But still, you could succeed. So it was, I knew that they already mentioned last time, 2012, I submit my application and I don't get it, I have to go away. So that was, and I was fearing for years, that when the chairman secretary is asking that the chairman wants to see me, that was not a good sign. And that's what I was told that, okay.
So I just want to remind everybody, you've got your cards, and if anything we've been talking about, you want to ask some questions, we're going to be talking a bit more. We haven't gotten to COVID yet, but just a reminder. So your boxes are out in the hallway. You've got to figure out what's your next act, and so what did you do then?
I mean, it was 2013. That Moderna was already get $240 million.
Moderna, the vaccine company.
No,
okay.
no, Moderna is a modified RNA that,
Right.
using our discoveries. And I thought that, maybe they change mind, and then I get a little red carpet event in Japan. I get money, and then I came back, and my stuff was on the hallway, and then they clearing my little lab there, and so chairman said, who's throwing out, this was not the chairman, it was a assistant, but she said that you have to talk to the chairman, and I told the chairman that, this laboratory will be one day museum. That's what I really said that.
This is going to be a museum.
That will be a museum, and he said, well, Kate, it might be, but now you have to move out here, and so that was it. And
that was it.
But I was not angry. That the neurosurgeon is doing surgeries. I didn't get the money, so I understand that. He just can look at, experts say what you are doing is no good, and what he can say. I was friendly. I mean, I run into him when I became famous, and I not run into, but just talk to him, and he told me that what, I am giving a lecture about you, about me, but it was like, he said, yeah, the title is How Did We Miss It? So it was, it's good. Yeah,
we have a saying in America, hindsight is 20/20, meaning like you can see things very clearly and calmly looking back. I mean, were you really that calm at the time?
Yeah, I have to say, I was calm and I was happy. And, I had this philosophy of, like, for example, when I was kicked out, I didn't start to say, why me? 24 years, I worked so hard, nobody appreciates me. No, I spent all of my energy, what next? I have to find a job somewhere. And running around a little bit in the country. And at age 58, I felt I'm the most successful that an old lady at age 58, I get 2 job offers, one from Moderna and from BioNTech. Vice president, yeah. That time, BioNTech had no website. We were just on the campus, but, I selected them.
Okay, so
it was, I was happy.
Okay,
Happy.
fair enough. So you start working at BioNTech, which was then a very small biotech company that was trying to develop vaccines with mRNA, and so for the next several years, that work is continuing, and then the pandemic comes. And I mean, I as a reporter was, reporting on these developments, and it was to me, it was really astonishing how fast things were moving. Did the whole experience with the development of a vaccine based on a lot of your work, did that take you by surprise? Did you think, or was this the day you were kind of expecting?
I have to say that BioNTech already signed up with Pfizer in 2018, well before the pandemic happened, and it was signed that it will develop influenza vaccine based on mRNA, and that was signed in 2018, and we started to work together with Pfizer. So by the end of 2019, we were ready for human trial to start. So we knew it was tested out, all of the assay, everything was done. But it was for influenza. But switching over to another protein is not a big deal. You have to have a different template, and the procedure is the same. Formulation was already set up. Everything was ready to roll. In these days when they said, okay, we need a different variants, another booster, and 6 weeks, and then you get millions of doses you can make. So it was already in place. It just you guys were not coming to the RNA therapy meeting, which we started 10 years ago. But if you would be there, you would know that it was already, other clinical trials were going, phase 2 trials were for heart failure and other things. So there were, and even for infectious disease, you know, that there were already rabies vaccine was developed, influenza, there were Zika in 2017, Moderna had 2 trials, CMV virus, different viruses. It was already trial, was already prior to 2020. It was just, that's what we have to do here, you know, that we should have told the public, you know, maybe they were not listening, but at least we should try to tell them, you know, that this is ongoing, so they would be not surprised that, oh, okay, pandemic, okay, but I know about the RNA vaccines is very quickly can be developed because already all of the experience.
So, I mean, and now this was a coronavirus, which is obviously different than the other viruses that might have been tested on. So, you know, in 2020, when you're waiting for the results, which were finally released in November, if I recall correctly, November 2020, were you holding your breath? I mean, were you?
No.
You knew.
No.
You were confident.
Yes.
Yeah.
Yes. You know, you have to also understand that, you know, that to know that what is important for a coronavirus, it was already, 1982 was the first conference on coronaviruses in Germany. So every year, those experts who are studying coronavirus, the scientists came together. They knew that what is on the surface, what is the immunogenic. So it was not a new, you know, on a way that they knew that the spike protein is what is outside. That's what you generate immune response. So many scientists is working on these things, and so all of this knowledge culminated that we can act quickly.
So, you got a mRNA vaccine in Philadelphia, is that correct?
Yeah, in December 2020.
December 2020. So, what was that like to sort of be getting your own vaccine back at?
Yeah, so they were setting up, you know, camera action. I was looking at the needle there, and I could see the vaccine in it, and I was thinking that I know the structure there, and I know some sequences there, and was, yeah, for me it was the biggest thing that we were walking there, and the chairman said people were waiting every 6 feet. This was, you know, the healthcare workers, and the chairman said, oh, these people, because with Drew Westman we were there, these people, you know, we sent them the vaccine they did, and these people who were waiting there, the healthcare workers, started to clap, and I started to cry.
So, what do you? I know that there's a lot. You know, that you had mentioned work on using mRNA vaccines for other diseases. I mean, right now, COVID is the only disease for which an mRNA vaccine is authorized or approved. Is that correct? I mean,
there is RSV and.
Is there? But there's a
respiratory system.
There's an mRNA one that.
mRNA.
Okay.
Yeah, it is Moderna. It was FDA approved.
Okay. So where else do you see, what other sort of diseases do you think mRNA, now that it's been showing its safety and efficacy in one context, where else do you think it will be put to use?
Yeah. So it is easy for me. I just came from Boston, the mRNA therapy meeting, and 650 people there, and the latest we could hear what is in the bench, what is in clinical trial, more than 250 clinical trial ongoing using messenger RNA. Most of them is advanced in a vaccine, viral vaccines, so they are developing for HIV and many other viruses that we have nothing, and some we have, and bacterial, for example, for TB, is human trial in ongoing, tuberculosis, and Borrelia, that's a Lyme disease causing bacteria. So these are all human trial, and malaria, this is also ongoing. So, and we can hear more about cancer vaccines. So those can be individualized, or there are known cancer components that, there are vaccines against this one. And also not just cancer is not just vaccines, but there are messenger RNA codes for antibodies, so that, which is already known that certain unique protein on the surface of the cancer cells, and then the antibody therapy is very expensive. So what is good, we didn't say about the mRNA, is affordable. It is very cheap because you make the medicine. It is just a blueprint what we injecting the mRNA. And so there are cytokines which help the immune system to rush to the tumor. So this kind of mRNA is development. There are genetic trials on, changing the genome for interrupting some toxic protein production by, that's Intellia developing. And many others, that mostly some acute disease and others. So there will be more. Definitely, there will be more. You will find out that more mRNA-based. What I mentioned, that one, imagine that in the refrigerator you will have a collection, and that mRNA is good for something, and use it.
So in 2021, Robert F. Kennedy said, the COVID-19 vaccine is the deadliest vaccine ever made. And Robert F. Kennedy Jr. has just been nominated by the president to be in charge of the Department of Health and Human Services, which is in charge of the National Institutes of Health, which is in charge of vaccine research, vaccine development, and so on. What is your view about the place of vaccines in American society right now?
So, this stoic view of what I followed all in my life, that
Stoicism,
stoicism, that, something, whatever is given, I cannot change, I cannot focus on. I focus on what I can change. And, whoever is the president, I really, I cannot change. So, it's this given thing. And I remember that many times I was driving on the Schuylkill Expressway, which is usually very stuck and something, and it influenced my life more, whether it is, some traffic jam there than whoever sits in the White House, the one way. And so I will just focus on things that I can change. And, I realized that we as scientists, we have not done our job, to talk to the people, to tell them that, hey, you think that this is the first time you get an mRNA injection? I mean, if you get year one, you were 1 year old or 5 years old, and you get the mumps and rubella, those are mRNA in it. Those are viruses, mRNA virus attenuated, and it even can replicate and then generate immune response. Those had mRNA in it, so that we did not describe, and everybody, oh, mRNA, and we as a scientist have a responsibility. That's why I am here, and you, so that we can help to the public to understand that and respect the scientists, because we worked day and night, and we want to have and the common good that everybody will benefit. We don't look at other interests.
Do you? We've talked about a lot of the hurdles that you had as a scientist, and it's that story reaches a point where you get a Nobel Prize. Mm-hmm. But when you look back at that and think about how science is done, do you think that there are ways to reform science so that these kinds of struggles are less of a problem for new generations of scientists?
I don't know. I am sure that more scientists can relate to my thing because, you know, they are working very hard and really they don't appreciate. If you read the book, you can find out that how you can be happy and everything is the environment is the same, but, you know, not stressed out and just to do happy life and despite all of this surrounding things. So I don't know what to do. You know that, I mean, modified RNA, so an investor would go there and say, oh, modified, that's great. You know, it won't happen. So I tried what it possible. So you are a scientist. You think that something is important. I formed a company, RNA-RX. No? Nobody heard. Yeah, because, you know, the patent we received for it, you know, the university gave it to somebody else. So without a patent, the company is not existing. So anyway, I tried my best so that if you believe what you are doing, spin out a company, and now as a company, try to convince, you know, investors and others that that what you are doing is important and move on and talk to bigger companies if you forward your research. So that's what actually is not doing well in Europe. That's why many of the patent is, you know, is not used there. But here is doing better, but we could do better and we needed talk to the people and we need to convince the society that what we are doing is for their benefit. We need to do that.
As I said at the beginning, if you haven't read the book, I would highly recommend that you do so, because there's a lot more to her life and her work than we could cover here. But thank you all for coming. Thank you to everyone online, and please give a hand to Katalin Karikó.